Good afternoon and good evening to everyone joining us from all over the world. Thank you for being here for today's webinar. This webinar is organized by the Chinese American Food Society, which was founded in 1975 and now has over 250 members. Today, we are excited to have three excellent female food scientists share their academic and industrial experiences on proteins for sustainable foods. My name is Huaru Zhou, and I'm an assistant professor of food chemistry at the University of Georgia. I'm one of the hosts for tonight's webinar. During the presentations, we kindly ask you to help us fill out a survey through the link that will be shared in the chat to help us improve it in the future. So now I'd like to invite my co-host, Dr. Zhang, to introduce herself and our first speaker.
Okay, thank you very much for joining us. I'm the other host for the seminar today. My name is Yi Zhang. I'm assistant professor of food science at the Pennsylvania State University. It's my great honor to introduce the first speaker today, Professor Jiajia Rao. Professor Yao joined the North Dakota State University in 2016. She is currently an associate professor at the plant science department. Prior to coming to North Dakota State, Dr. Yao worked for PepsiCo for 3 years. She obtained her PhD degree in food science at University of Massachusetts Amherst. Dr. Yao has published more than 100 research papers and secured research funding from different funding agencies. Today, she will be presenting on K plant ingredients for alternative proteins, opportunities and challenges. I would like to tell the audience that please feel free to type your questions in the chat box during the talk, or you can use the raise hand function at the end of the talk if you prefer to interact directly with the speaker. Okay, please join me in welcoming Professor Yao. The floor is yours.
Thank you. So I gotta share my screen just to wait a second before I start talk. Let me just. So can everyone see my screen very well?
Yes.
Okay. So thank for Dr. Zhang's nicely introduction, and thanks to everyone for attending tonight's or morning's presentation. So today I'm gonna talk about my past research or current research on the key plant ingredients for alternative protein by using the pulse protein as examples. So as everyone know, be aware of that, so now the plant protein became the buzzword. A lot of food companies also would like to incorporate plant proteins and all different types of food. That's why I also search up online to find, okay, the plant-based protein global market report is continuous growth with the growth compound like 17.9. Maybe later on the third speaker can correct me if that data is wrong. And as you can say, so all the plant protein fortified food, so they can come from different sources. For example, the plant protein ingredients from legumes, and can be from the nuts and oil crops or cereals. And so even though the plant protein is buzzword, but we still met a lot of challenges. For example, like unpleasant flavor, like pulse protein usually has some bitter flavors that if you incorporate this plant protein, you might get some unpleasant flavor and sensory aspects. And also some of the protein has a limited functionalities, depends on the protein sources. And in terms of nutritional quality, majority of plant protein cannot be considered as a complete protein. That's why also made the challenge in order to incorporate into the food system. And I would say for the past couple of years, and our group has been already work on the different type of plant proteins, like from the cereals and from the oil seeds, and majority of the protein is from legume type of protein, like different type of the like pea, yellow pea, green pea, and chickpea, lentil, as well as some beans. And today I also just use pulses like pea protein as example. So in general, so the protein cannot be only considered as a nutrient content that can be incorporated into the food system, but you also need to consider, let me just move the part, and the functional property that can be used in the food system. For example, if you apply the plant protein into the beverage, you need to consider about the solubility. And if you apply the protein into some different other food system, you may need to consider about the emulsifier, for example, foaming properties, as well as the gelling properties. And so majority of this function is governed by the protein's molecular interaction. The molecular interaction can be strongly impacted by many factors, such as raw materials, what type of materials you use, and the different isolation methods, as well as some modification methods. So today I'm going to give you some simple examples of how the different raw material, as well as the modification methods, to impact on the protein's composition, as well as their functional properties. So as I said, I'm going to use the pea protein as example. So let's take a look at the general protein's composition derived from the pea, yellow pea, for example. So the pea protein, the two major protein composition we consider as the salt-soluble globulin and water-soluble albumin that account for 80% and 20% respectively. In terms of the salt-soluble protein, so it can be subdivided into two major categories. One we call the legumin, another one we call the vensin. So as you can see, based on the structure and molecular weight, you can see it's much larger as compared to dairy protein. So that's why you met some situation in terms of solubility. And in terms of the second structure of the protein, the majority of the plant protein, like for example, the pea protein, is considered a large amount of beta sheet and followed by a certain percentage of alpha helix. So for the beta sheet, for example, so the oxidized flavor compound, like unpleasant flavors, can be trapped into the very rigid structure, beta sheet structure. So that's why. So it will extract and return the unpleasant flavor towards end of the product, even though through the protein extraction system. So here I just give you an example how the different raw materials impact on the protein composition as well as the flavor profile. For example, so in the past we extract protein by using the pea cultivars. For example, the widely used agaza and spider and chimpanzee, as well as some of the North Dakota's child protein. So as you can see, after extract protein, we measure the functional property. We found that we just use foaming property as one of example. We found that so by using different pea cultivars, the functional property is different. But why the functional property is different by using different raw materials? It's all from the pea, but just use a different cultivars. So what we found, the different cultivars extract isolated protein from the different cultivars, for example, and it contains a different protein composition, such as legumin to vensin ratio. To further confirm this, so we get conclusions. Pea cultivar with a higher vensin content shows a better foaming capacity. To further confirm that, we also separate the different subunit from the protein, the total pea proteins, for example, the globulin, legumin, and vensin. So we also confirmed that. So the higher vensin content has a higher associated with higher foaming capacity. And in the meantime, we also measure the whole flavor profile of the pea protein isolate from different cultivars. And if we use the flavor marker as example, so we can find that so the concentration is quite different based on the flavor marker from the different cultivars. So that's we would we can give the information to everyone. If we use the different raw materials, it might show the different functional properties and as well as a flavor profile. Even though we can select, okay, in this case, you might wondering, we can select the premium cultivar, premium products by using optimized operation conditions, extraction conditions. Can we get the very good functional property and also the flavor profile of the protein product, plant protein product? So, but it seems we're still not satisfied. That's why for the past decade, a lot of scientists and also researchers start to explore the different strategy to improve the functional properties, for example, like physical, chemical, enzymatic method, as well as breeding program. So in the past, our lab also explored a different physical method to improve the functional property of the pea proteins. So in here, I just give you one case study by using the ultrasound treatment for improving the solubility of pea proteins. So here, just we randomly select one of the commercially available pea protein isolate, and we also measure the protein composition in the approximate analysis of the pea protein isolate. So what we found is the protein is around 80% protein content and still contain 10% carbohydrate and as well as some ash and moisture. And here, so let's explore. So maybe some of people has already been aware of that if you have some experience by using the pea protein isolate. The pea protein isolate solubility is really low, but how low? So in here, we measure absolute solubility by adding the 2.5 grams of the commercial pea protein powder mixed with 25 milliliter of water system at pH 7 and a centrifuge. So we just measure how much solid content on the superlatent as in here. So we consider as a water soluble fraction. So what we found only 0.18 grams out of 2.5 grams of pea protein isolate that powder can dissolve in 25 milliliter of the waters. So in this case, as many researcher explored, so the ultrasound could improve the solubility of the pea protein isolate, and many researchers believe that because they changed the structure or property of protein, that's why they could improve the solubility of pea protein isolate. So we buy this idea partially, but we also would like to do some simple experiment to say how they can improve the solubility of protein by designing a very simple experiment. We call it sequential high intensity ultrasound treatment. In here, so we still add the 2.5 grams of PPI into 25 milliliter of the water system, and then treat the ultrasound treatment at 100 watts at a certain time. And after that, so we centrifuge to collect the water soluble fraction here. And then for the water soluble fraction, we continue to treat the ultrasound treatment by increasing intensity. And after that, we also add back to the water to continue use the same sample to collect the water soluble fraction and separate until there's nothing you can do until you precipitate. There's no more precipitate anymore. And so in this case, we call the sequential high intensity ultrasound treatment. And so let's discover the very interesting results. So what I found, so like I mentioned before, so we measure the total mass of the superlatent fraction. In this case, as the ultrasound treatment, intensity treatment increased, we found the solid content, the total mass in the water soluble fraction, which means solid content in the water soluble fraction, gradually increased until reach the plateau, and later on decrease. So we also measure for all the soluble fraction, it's all protein, or does also contain other ingredients. So in that case, we also measure the protein content in the superlatent part for each ultrasound powder treatment. What we found in here, so you see, initially it contains only 50% of protein, and with ultrasound powder increase, the protein content increase, but it's not reached 100%. Until it reaches plateau, it no longer reaches. So which means it still contains other ingredients in the water soluble fraction. It not only contains the protein, but what are they? Think about, we mentioned about in the past, the commercial pea protein isolate, it also contains a larger amount of carbohydrates, contains 10% of carbohydrate. So in this case, we also measure the total dietary fiber content in the water soluble fraction, as you can see from previous picture, which means it's a superlatent phases. And then you can see that, so this trend, as ultrasound powder treatment, the intensity increased. So the trend of total dietary fiber content also matched with previous protein content. So which means, and also reach the highest point when 150 of ultrasound powder treated to that protein solution. So which means, so not only the insoluble protein migrate from precipitate phases to the superlatent phase, the dietary fiber also converted from insoluble fiber to the soluble fiber. And we also did the research on how the ingredients interact with each other. So we found out they are not stayed in the superlatent phase individually. They found some complex. So that's why, based on the confocal and the TMN results, we found that. So that's why we proposed a new mechanism. So the ultrasound treatment could not only improve the protein solubility by changing the protein structure, but also will help to promote the fiber convert from insoluble fiber to the soluble fiber and form the soluble complex between the protein and polysaccharide. So that's one of the examples to showcase how can we use the ultrasound treatment to improve the protein's functionality, but at the same time, you also need to consider about the minor components in your protein samples. It also will respond to your treatment. So in addition to the physical methods, our lab also explores the different chemical modification methods. Because of the limited time, so today I only share what's one of the methods we call the phosphorylation. So in general, so the phosphorylation is to bring the phosphorate to the protein molecule, and usually they react with serine residue on the PPI, and also with improved functionality such as solubility as well as the surface property associated with solubility. But in the past, most of the time, the phosphorylation, the method, because they usually requires the hash chemical reaction condition. For example, the high temperature with the large amount of phosphorate required to react with protein, or use enzymatic method to improve the reaction efficiency. But this is not adaptable for the manufacturing, the technology. So in this case, we try to propose, can we use some of the manufacturer adapted simplified method in this to form the phosphorylate the pea protein isolate. So that's why we propose to add one of the emulsifier salts during the protein extraction processing towards the end. So you can see at the precipitation phase. So we add the emulsifier salt and cold spray dry emulsifier salt with isolated protein solution and get phytomodified pea protein isolate. So in here, in order to maintain the protein content at 80%, in that case, we still can call this product as a protein isolate rather than lose the isolate label. So that's why we also give the very low emulsifier salt concentration to bring very low emulsifier salt concentration into the pea protein powder. So in here, we use two ratios. One is 95:5. We label as a L, which means a low concentration one. And another ratio is 90:10. So we label as a high concentration one. So let's take a look at the results. First of all, let's take a look at the results from the FTIR. So we also would like to say is that emulsifier salt has already successful to bring to the protein molecule. So based on the new peak at the 879, the wavelength, so we can say this is a stretching than POP. So that's why we can confirm that the phosphorate ester was successfully introduced to the PPI molecule. And in terms of the second structure of pea protein, modified pea protein, as we can say that, so the beta sheet was gradually decreased and the alpha helix is slightly increased. And we also measure the foaming and the emulsifier properties. And as we can say that, so all the emulsifier properties, including the the particle size as well as stability increased, and the foaming capacity and stability also increased as well. But we don't see the significant increase in terms of the protein solubility by using this cold spray dried method. And so because it was a limited time, so I would like to wrap up here. So, and I would say, so there's a gap between by using different to understanding the how can we apply the different plant protein in the final applicate product, for example, the beverage or bakery goods. So we need to understand what kind of raw material have been used and what type of the isolation method they have been used in the past, and what is protein composition. So these parameters are going to definitely impact on the functionality and also ultimately determine the final application area. And so I also would like to thank everyone to listening for my presentation. And finally, I also would like to promote one of the great artists of building. So in last year, so our program, a food and safety research program, moved to the new state-of-the-art building. So this is that's why I posted some pictures, including a lab and new sensory kitchen. So if you are interested to visit a lab, if you by chance to stay around the North Dakota or Minnesota, just let me know. I can give you a tour for our new buildings. So that's all I have today. So if you have any question, I would like to take any question.
Thank you very much, Professor Yao, for the wonderful talk. And congratulations to the new facilities. It's pretty exciting. Okay, let's open the floor for questions.